welcome to this series. Here we will be understanding two different cases of electronic system that can be used for screening the oral cancer patients. So, there are two ways of screening. One is imaging, where you put the probe in the mouth, take the photo of the suspected region and send it to the clinician and get the the advice whether the person should go for histology, where you have to remove the tissue, right. Second one is cytology and histology. Cytology is where you have to take the cells from the oral cavity, and those cells are tested by the oncopathologist, and if the cells are atypical, the result comes as a atypical, or the person is okay. If the cells are atypical, then the person has to go for histology, where the tissue is taken out and further gold standards are using the gold standard. The patient is given a diagnosis whether a patient is suffering from a cancer, or you can say malignant tissue or a pre-malignant tissue, or it is a benign tissue, right, or the patient is okay. So, if patient is okay, we do not have to worry, because that itself in the screening stage we can know. So, now the point is, let us see how the change in the cells would help us to understand whether a person is susceptible or has a chance of having a particular disease. This disease we are talking about is a cancer. So, his chance of having a cancer, all right. So, it is very sensitive and very crucial to understand the change in the cell morphology. Now, there are already worked around this area. So, we will be talking about how the cells will look like, and what is the cell morphology, and how what are the present way of understanding the morphology of the cells, and then what can we design as an engineer to a system that can help us to screen the patient in a effective, cost effective and a rapid manner, all right. So, the topic today is to understand the cell and tissue morphology, and in that let us first understand the cell morphology. Now, if I talk about cell morphology, the definition of the cell morphology would be to study, or is the study of the size and shape of the cell, right. Diversities. So, what are the cell diversities? Cells are diverse in terms of shape, size, internal organization and functionality. When you talk about size, so most of the cells are microscopic in size, right. And what is the purpose? Purpose is to understand the functioning and the pathogenesis associated with the malignancy. Malignancy is the possibility of cancer, all right. Significant of morphological cell analysis. Here the abnormality identification and classification, such as early cancer detection. You see, this is the extremely important part where we have to focus. Significance of morphological cell analysis. Here the why we have to understand cell morphology, because cell morphology, if there is a abnormality in the identification, that is there is a change in the shape, change in the size, a change in internal organization, right. There are double nucleation, lot of other stages are there where we can understand that there is something wrong with the cells, and that is why we can help for the early cancer detection. It can help in early cancer detection. Now, you can see three different photos. One are skin cells, you can see, then there are nerve cells, and finally there are bacteria cells, right. Now, bacteria is an enemy to human cells in general, but of course, there are small quantity of bacteria present in our body itself, which are good bacteria. Okay. We are talking about the bad bacteria. So, certain bacteria are good, certain bacteria are bad. If it is within the limited quantity within the body, it is useful, above it is harmful. One example is E. coli. One example is E. coli. So, what are the methods in morphology? If you see, we can divide into two different techniques. One is the chemical technique. and second is microscopic techniques. So, if you talk about chemical techniques, then there is histochemistry, right, and in histochemistry differential staining of cells using dyes, methylene blue, right, malachite green or safranin. These are the dyes that are used in histochemistry to understand the change in the cell morphology. Then by choosing appropriate dyes, the histochemist can determine the acidity alkalinity of cells, also locate the specific enzymes. All right. While we talk about microscopic techniques, then microscopic techniques, the histologist and cytologist use microscopic techniques to study the structure of living cells, right. And what are the structure of living cells? We will talk in detail in the following slides, but what are the techniques or what are the microscopic techniques used? Then we can see that there are 6 different microscopic techniques used, right. One is the SEM, which is scanning electron microscopy, then second one is light microscopy, third one is phase contrast microscopy, fourth one is interference, then we have fluorescent, and finally we have polarization microscopy. All right. So, there are 6 different microscopy. Now, let us see each of those in detail. So, the good thing is I have videos for you. So, you can go through the videos and it will be easier for you to understand, right. There are short videos except one microscopy. So, it will not take much of your time when you look at the videos. So, let me play first the scanning electron microscopy. Okay. Now, as you have seen how the SEM can work, right. SEM can not only be used for understanding the cells, but it can also be used to understand the extremely small structures in case of the MEMS based technologies. We have seen the SEM images in our early modules, right. So, but SEM can also be used to understand the cell morphology. Let us see the another microscopy, which is phase contrast microscopy. Let me play that video for you. So, phase contrast microscopy. Okay. So, phase contact microscopy you have seen now, right. Now, there are several other microscopy where you can understand the whether it is bright field or it is fluorescence, because in some cases we are staining with HNE and we are okay with bright field. If there is a DAPI and there is a Catherine blue, or there is a red dye, or there is a green dye, right, which is so there is a FITC, and then there are a lot of other nomenclature given for this particular dyes. So, the point is, or names given for the dyes, and these dyes show a particular color when you look under the fluorescent microscope. So, we will also see how the bright field microscope looks like, and then we will see how the fluorescent microscope looks like, right. So, let us see the video of bright field and fluorescent microscope. I will first play bright field.
Hey everyone, quick biochemistry basics here. Let's talk about bright field microscopy. Bright field microscope is the most common and simplest of all optical microscope. A typical bright field microscope has many components that includes a light source, condenser, stage, objective, eyepiece, rough and fine adjustments. The light source can either be a halogen lamp or an LED. The condenser collects the light from the light source and directs the light to the specimen, which is kept on the stage. The light passing through the specimen is collected by the objective lens, and the final magnification is created by the eyepiece. The total magnification of the specimen observed is the product of magnification of the objective and the magnification of the eyepiece. Now let's talk about objectives. The objectives are the most important imaging components of the optical microscope and play a key role in magnification and resolution of the specimen. All the objectives have an information written on it. This includes the name of the manufacturing company, magnification and numerical aperture, the term plan is used to indicate corrected field curvature. Field curvature is a common aberration caused by the spherical surface of a convex lens. In this phenomenon, not all parts of an image are clearly focused. While focusing in the center of an image, the peripheral region gets blurred, and while focusing in the peripheral region, the central region gets blurred. To avoid the problem of field curvature, modern objectives are designed to give a clear, flat image, with no blurring in center or the edge. The objectives with corrections in field curvature are called plan objectives. Corrections for chromatic aberrations. Chromatic aberration is a phenomenon in which the lens fails to focus all the colors of the light at the same point. It occurs because the lens have different refractive index for different wavelengths of light. The term achromatic lens means it is corrected for two colors, red and blue. The term fluoride or semi-apochromat means the lens is corrected for three colors, red, blue and green. And the term apochromat means the lens is corrected for four colors, red, blue, green and infrared. Bright field microscope typically has low contrast as most of the biological samples transmit most of the light. To increase the contrast, staining is often required. The disadvantage of staining is that most of the cells are killed and live cell imaging is difficult.
Now, since you have used the bright field and seen the bright field microscopy, let us also see the fluorescent microscopy, and let me play the fluorescent microscopy as well. So, I will as a part of this course we will show it to you how the bright field microscope works like, right, and we will also see inverted microscope, we will see a stereo microscope, we will also see a metallurgy microscope. So, we will see 3 different microscopes to understand the how the cells looks like, how the devices looks like. Okay. Now, then the even complex microscope comes as a polarization microscope and interference microscope. So, let us see the videos of polarization microscope as well as interference microscope, and then we will go to the how can we do the biomedical research when you understand the cell morphology. Okay. So, let me play both the videos. First I will play polarization microscopy followed by the interference microscopy. So, I am playing polarization microscopy now. Okay. Now, since you have seen polarization microscopy, let me also play the interference microscopy. Alright guys, so what we understand? We understand there are chemical techniques and there are optical techniques or microscopic techniques. We have seen 6 different microscopes, and here in each of those videos you were able to understand how each microscope would work for and how we can use it for so this all 6 microscopes can be used for understanding the cell morphology. Now if I want to further understand what are the research components or biomedical research components, if you want to understand the analysis of the cell morphology, then we can see that the applications are in to understand the segmentation and analysis of histological tumor sections, boundary detection of cell nuclei, morphological characteristics, analysis of specific biomedical cells, understanding the chemotactic response and drug influences, identifying cell morphogenesis, indifferent cell progression, morphological features, quantification for grade cancerous or precancerous cells, and finally feature extraction and texture analysis for automated cell classifications, right, or classifications of cell. So, there are several applications if we understand the morphology and we want to do the analysis, right. So, now to understand this, right, let us see how what can we do. So, before we go to that the way it is carried out, the current technique that we use to screen the patient, let us first understand a technique called cytospin. Alright. Before cytospin, let me draw a slide so you understand why we have to go for cytospin. Now generally when the cells are taken from the mouth, okay, the cells are taken from the mouth with the help of a cytology brush. When it is taken from the mouth or oral cavity, it is placed in a solution called phosphobuffer or PBS. So, there are cells in PBS. These cells in the pathology lab, they are smeared on the glass slides. They are smeared on the glass slide. So, how the smearing works? You take the cells, put a drop here and then take another slide and then you push the slide or smear the slide across the bottom slide. What will happen when you do that? When you do that, the cells will be, it gets spreads like this. This is called smearing. Alright. The disadvantage of this particular technique, is there is a double nucleation. There is a the cell will get, it will be clumped. Another way we can say it will be clumped, right. So, it is very difficult to identify the boundary of itself and it will not be uniformly distributed, This is with the help of smearing the cells, smear, SMEAR, right. You understand, when you take the cells from the oral cavity, you place in the along with PBS, and when you take it out and you smear it, then you will find the clumping of cells, and clumping of cells will not help the oncopathologist to clearly identify whether there is a change in the morphology of the cells or not. So, to avoid that, we can use a system called cytospin. I will discuss in detail about cytospin in the next module, but let us see right now how the cytospin works. Okay. Cytospin centrifuge used to study the morphology of cells. The centrifuge spins and flattens cells onto a glass slide. Extremely it improves the cell yield in samples and ensures high diagnostic efficiency. Also, it is cost effective with better preservation of cell morphology and effective approach to determine the malignancy. Alright. So, we can see this particular cytospin centrifuge system, and we have the video for that as well, and then you can see here the loading system for the cells, and you will load the system in the cytospin so that you can load the glass slide here and the cells here. So, at a very high speed the cells will be smeared on this glass slide, and the advantage will be by using this technique you can have you can preserve the cell morphology and you can have a uniform distribution of the cells. Alright, there are these two advantages. You can preserve the cell morphology and you can have uniform distribution. Now, why it is important to have uniform distribution? Because if I have uniform distribution of the cells, right, then I can identify whether the morphology of this cell is different than this cell, or all the cells looks alike, right. Also there are a lot of things that we have to understand: double nucleation, cell to nucleus cytoplasm ratio, and whether the cell shape is changed, right. So, a lot of things are there that we have to take care when we are understanding the morphology of cells. So, the spinning on the glass slide with the help of cytospin will help us to preserve the cell morphology compared to the smearing. Now, how to use cytospin and why cytospin we have to use it for understanding or for delineating benign and malignant cells, right. Why it is very important. Now, I already told the importance of that the importance is so that we can understand the cell morphology, right. Cell morphology is so that we can understand whether the cell shape is changed or not. Now, the diagnosis disease like cancer at looking at the cluster of cells is a help of cytology. Cytospin is a cytology method that is specifically designed to concentrate cells that are found in a small numbers. Now, there are two things. One is a cytology which is cytospin based. Cytology is the study of cells, by the way. Cytopathology is study of the cells, whether there is a change in the shape or morphology from the pathological angle. Cytospin is a system that is used to smear the cells on the glass slide, right. So, cytospin is a cytology method that is specific to design and concentrate the cells that are found in small numbers. Now, if you see here, you see there is two system, one is the centrifugation and then another one is the supernatant. So, see in this particular case, let us say if I have a blood and I want to separate the blood cells, then I will use the centrifuge. So, centrifuge is that it will put the cells in the bottom, and the red blood cells would be at the bottom because they are the heaviest one, and you will see the layer of the cells, and finally, there is a supernatant. So, in general, if you have just cells in a PBS and you want to wash the cells multiple times, you can use centrifuge. What will happen that whenever you wash the cells, right, and you put in this particular device, then what will happen when you centrifuge it, this cells. So, let me just draw it correctly, help you to understand properly, right. So, here the cells are everywhere along with the PBS solution. When you do centrifugation or when you centrifuge the cells, then the cells would be at the bottom and the top will be supernatant, and that supernatant we can discard it, we can throw it out, alright. So, because this we do not require it. So, when you have to wash the cell multiple times, you can use centrifuge. When you want to smear the cell on the glass slide, you have to use cytospin, alright. Cytospin also works on the centrifugation technique, but the system is such that the design of the cytospin is such that it will smear the cells on the glass slide instead of just separating out like supernatant and the cells at the bottom, alright. So, let me play these two videos and you will understand the things in detail. Anyway, like I said, I will tell you the importance of cytospin in my next module, you will understand it better, alright. So, let me play this first video.
제작 시범 보이겠습니다. 옆에 먼저 옆으로 한번 보여주세요. 옆으로도 한번 보여주세요. 네, 됐습니다. 그 다음에 검체를 깊게 돌려주시고 1800rpm에서 10분간 돌리도록 하겠습니다.
Right? Now let me play the second video also.
This is the L'Oreal WS650HZ spin coater. It has a maximum rotational speed of 12,000 RPM. The controller allows operator interaction in real time during the process execution, including pausing time, stopping, and continuing on from that point. The closed bowl design allows most coating materials to dry in a quiescent state, increasing uniformity and minimizing particle contamination. This convenient tabletop unit is powered by 120 volts AC, single phase. It also requires vacuum to be at least 15 inches of mercury or more. First, I'd like to give a demonstration of the speeds. For that, I'm going to use our little timing strip. First, this is the compact control panel, completely self-contained. I'm going to call up a program here, program 4, and what we'll first do is we'll change the spin speed to, let's say, 4,000 RPM. There we go. And then first we will apply a vacuum. It tells us that we are ready. And we'll go. Okay, when it's done, in order to go on and run another program or any other process, you must open the lid. This signals it that it's ready for another run. What I'll do in this one here is I'll double that to, let's say, 8,000 RPM. And we will repeat. Lid. We're done. It'll always say done, and after you lower the lid, as long as your vacuum is satisfied and your nitrogen purge, it'll tell you that it's ready. We'll do one more spin, and we'll do this at the maximum RPM of 12,000. So, one, and go. done. Next, we're going to spin a small wafer. It has this very, very nice little adapter here that you can fit right on top of the standard chuck. First, we apply vacuum, and we'll spin this little wafer at the maximum speed. Now next, I have this chart here, and this is wafer size versus spin speed. Obviously, the smaller the workpiece, the faster you can spin, and they've given a little guideline here. First, we ran this little guy up to 12,000. Now we're going to run a 100 millimeter wafer, as suggested, up to 6,000. This is actually 150. We use a little 100 here. Apply a vacuum. And we will also edit that speed down to 6,000 RPM. And here we go. Done. Turn off our vacuum. Move our wafer. Now, the spin coater has the capability of actually being able to spin a full 200 millimeter or 8-inch wafer. However, it is to be noted that in order to do so, you would have to remove the top and take off this plastic splash guard, because that just isn't enough room for it. But it actually has the capability of spinning a 8-inch wafer. However, what's important also in this particular unit is that it has the capability of spinning a full 5-inch by 5-inch square substrate. I have a piece of glass here that I can use to demonstrate. You center that respectively. And even though it's about approximately 7.5 inches across here, if you look through the top, it still comfortably fits in between the splash shield. Now I'm going to spin this one. Since this is closer to the very larger size, which they recommend between 2.5 and 3.5 KRPM, I'm going to spin this at 3,500. And here we go. vacuum first. There we go. Ready, lights on. Start says yes. And there we have it. Very versatile, very compact, lightweight, fits into anywhere, and very easy to use.
Okay. So, this will be the end of this particular module, and in this module like I said we are just understanding the morphology of the cells and how different techniques are used to understand the cell morphology, right. And in the next module we will look in detail how the cytosine system works, and along with the cytosine system we will also talk about how the characteristics of the cancer cell helps us to understand whether the characteristics of a cells helps us to understand whether the cells are from normal region or are from the cancerous region, alright. So, this is the last slide for this particular module, and in the next module we will talk about the remaining stuff along with the details about cytosine system, because I want to teach you how exactly cytosine system looks like and why we want to go for cytosine. So, I will make a PPT for you so that you understand the importance of cytosine, alright. But for now just go through these videos, these are more like informative videos so that you understand how the system works like, right. But again to understand the cell morphology and to delineate the cells based on the cell morphology is our goal, right. But and from that we will understand whether the cells are atypical, that is it has changed the morphology, or they are normal, alright. And if they are atypical then the patient is asked to go for the histology. So, we will talk that thing in the next module. Till then you take care, I will see you in the next class.